My name is Corey Padilla. I head up our scientific affairs part of Dovetail Genomics. And for the past year, what that's really meant is diving into how do we compute proximity ligation data, and a part of that being HiChIP data. So what we're going to do is we're going to spend a little bit of time, actually probably a decent amount of time on the introduction, getting everybody on the same page. Because there's a lot of definitions here that we all need to be kind of on the same page about, and they kind of cross over between ChIP-seq and HiC. So there's some specific terminology that we'll need to all keep in mind as we move forward. Then we're going to talk about calling significant interactions, not calling loops. And I'll tell you why I don't use the term loop coming up here in a few slides. And then we're going to show you how to plot these contact arcs that are kind of hallmarks of HiChIP data in two different ways. In R, using very simple commands with a very common bioconductor package called sushi. And then I'll tell you about how to format the data to plot in the epigenome or the WashU epigenome browser. Then finally, we'll end on 1D peak calling with HiChIP data. One, how to do it, and then also things to keep in mind if you are to use HiChIP data in calling one-dimensional peaks. And then I'll just end with some general things to keep in mind as you move forward. So before we get started, if you're going to follow through this workshop tutorial, whatever you want to call it, later on, the things that you should have are your alignments in BAM format. You should have your valid pairs files in .pairs. And then you should have experimentally derived ChIP-seq data. You know, these might be able, you might be able to grab these from ENCODE. You might have these from other previous ChIP experiments from your example, but we highly recommend having ChIP-seq data to ask the questions about this interactome data type, as long as it's anchored over a DNA protein binding site. All right, and then here's just sort of the general landscape that we'll sort of be walking through today. I'm not going to go through every single one of these, but we're going to sort of, you know, compute coverage. We're going to look at one-dimensional peaks, and then we're going to ask what does the interactome look like over those one-dimensional peaks, and then how do we plot that three-dimensional data type. So the name of the game when you're dealing with HiChIP data is you're trying to boil down this contact matrix type approach to put p-values on these interactions and then plot it like this, where all of these arcs that are connecting parts of the genome all have are all interacting, and that interaction has a p-value that is significant, right? That is really what we're trying to do here when we analyze HiChIP data. And so that's what we're going to do. And so in order to do that, let's go through some kind of basic definitions to get everybody on the same page. So first off, what are contact arcs? So the way this works is you have HiChIP chimeric read pairs, where you have region 1 of genomic DNA that's captured by a chimeric junction, where at that junction there's a biotin, and then the other side is another piece of genomic DNA that's far enough away that's representative of proximity ligation. And so what we do is we map these reads to the genome, and they fall out like such. And then we bend the genome, so we kind of bucket the genome into these bins. And then we draw arcs between the different read pairs that fall in different buckets. And then we combine and count the arcs that have shared bin pairs. Right, so notice there's 3 reads here that all fall within this bin. However, we're only combining the red and black, combining and counting the red and black ones here because they have the same pair, where this orange one has a pair combination that is different from the other 2. And then once we have the pair or the interactions between 2 bins and the counts that occur between those 2 interactions, then we can just go ahead and scale the arc height by either distance or by contact frequency. How many counts do you see that connect any two bins? That's great. So how does this compare to a contact matrix? So what we'll show you today is if we take this data and go with the R wash U route, and you can plot these arcs out like this, or you could take this data type and use juicer cool tools and just print out a contact matrix like you would with HiChIP data. But these two are equivalent. These two images here are the exact same, where between bin 1 and bin 3, we see a higher contact frequency as we do in this contact matrix, and then between 1 and bin 3, 4, 5, 6, we do see a contact linking them, but it's at a lower contact frequency, so it has fewer contacts here in the contact matrix. So these things, all things considered, are held equal, but it's easier to kind of place p-values and filter out these two interacting bins in terms of arcs, as it is sometimes to go the contact matrix route. So the next thing that we want to consider here is that HiChIP data is made up of primary and secondary peaks. Now, primary peaks refer to the DNA protein binding site. So when you do ChIP-seq, just normal, regular old ChIP-seq, what you're getting is the primary peak, right? Where that protein of interest binds to the DNA, right? And that's where you see this enrichment. Now, when you're using HiChIP data, or make a HiChIP library, what you're asking is where's my primary peak? And then what are the interacting regions that are interacting with that primary peak? And so oftentimes what you see are these secondary peaks that occur away from the primary peak that are reflective of regions of high interaction. And so if we go back to this sort of, read pair approach, the way to think about it is one of your read pairs will always have an anchor or will always be anchored to a ChIP primary peak footprint. The other part of the read is free to wander about with respect to proximity ligation, right? And so we can see here that there are two reads here stacked far away, and so that's why we see a slightly larger peak here closer to the primary peak and a smaller peak further away. Right? And so when you're asking questions of these data, you kind of have to keep in mind here that if you don't have ChIP-seq data, it's really hard to tease apart what is a primary and a secondary peak. But if you do have ChIP-seq data, it's really easy to know because wherever you have a ChIP-seq peak, that's a primary peak. Any peak that falls outside of that ChIP-seq signal in the HiChIP data is considered a secondary peak. Okay, so we're going to use this like primary peak, secondary peak, and these arcs, these interaxomes, to call significant interactions. Now, these are more commonly referred to as loops. And when you're thinking about HiChIP data, you're thinking HiC data, you're thinking, CTCF mediated loops or enhancer promoter loops or promoter loops. That's not necessarily the case here with HiChIP data. Loops are just significant interactions that are anchored at a protein binding site. And the interpretation of these interactions is very much dependent on the protein of interest. So if you think about something like H3K4 trimethylation, which is active promoters, when you conduct HiChIP on active promoters, what you're asking is, what are the regions that are interacting with in active promoters? So significant interactions, the loops that you get, are really just what regions of the genome are interacting at a higher frequency with active promoters than you are with the rest of the genome. Now, when you're doing something like CTCF HiChIP, you are in fact calling loops. Right? At a larger scale, you're calling tags, but at a smaller scale, you're calling CTCF mediated looping events. Now, if you're using something like H3K27 acetylation, which is an enhancer mark, you're simply asking, what are the regions that are significantly interacting with my enhancers? And so a lot of people think of these loops as you're making this nice circle that are anchored at a cohesion event, but that's not always the case, right? You might have some like DNA folding where things are being kind of pulled and roped across each other. You might have things like loop extrusion going on. So There's a lot more subtleties in the HiChIP data than there are in the HiC data. It's not that the HiC data doesn't have those, they just don't stand out as much because there's a lot more background noise, or there's less signal to noise in HiC data than there is in HiChIP data. Okay, so now that we know what significant interactions are, how do we define a significant interaction? Well, it's not like a coverage signal like you would do with ChIP-seq. There are two things you have to consider. You have to consider what the distance is between two interacting bends, and you have to consider what the contact frequency is between those two bends. So, and what we typically see with almost any proximity ligation data is it follows this logarithmic decay, where at shorter distances, there is a higher contact frequency. And as we extend out, that the frequency decays with distance, right? And so when we have these data, right, we can model out that curve. So if we take these data points, we can draw out a line of best fit or model, right? And which is a logarithmic model. And then what we can do is taking our empirical data and our model, we can put some confidence intervals on that model. So now what this means is you now have at any given distance, you have a confidence interval of what the contact frequency should look like, right? And so then you want to ask the question of my raw data, what interacting bends fall within my confidence interval and what bends fall outside of my confidence interval. And ones that fall outside of the confidence interval, those are significant. Those that fall within are not. And so it's this approach here where you do kind of have to build that logarithmic decay model to be able to ask the question of any two interacting bends, what's the p-value that the frequency is higher than expected, right? And so again, this model is empirically derived from each sample set. This is not like a observed versus expected. This is you're taking your HiChIP library, you're modeling out that curve, you're building the model, and then you're putting the confidence intervals around, and then you're asking that question. Another really important thing here to keep in mind is this model will look different for different antibodies. So here I like to think about extremes. Think about CTCF, things that act upon large distances, right? So you're gonna have a higher contact frequency out at these larger distances. But then if you go to the other extreme, like maybe pol 2, where you're really binding to the promoter site and then elongating over the gene body to transcribe, you're gonna have a lot more short interactions. And so that's why you use your sample-derived data to actually build this model, because it's going to vary dependent on that. The other thing that this model is going to depend on is what type of interaction you consider. Now, the next question you're going to ask is, what do you mean by different interaction type? So let's define those interaction types. So here is our viewpoint. Here is our primary peak that we know from our ChIP-seq data. Here are all the possible interactions from the raw data that we get from the HiChIP library here in gray. So now there are a few things that you could consider when you're selecting your interactions. Are they going to be interacting with only peak to peak, right? Meaning that read one has to fall within a bin that aligns with a ChIP-seq peak. The other bin pair also has to fall under a ChIP-seq primary peak, right? And so you're really just narrowing your question into, out of all the peak-to-peak interactions, which ones have a higher frequency that's outside of my modeled curve, right? Then there is peak to non-peak, where you consider all the interactions except for ones that occur at another primary peak. So basically, you're looking at all the interactions except ones that fall on a paired bend that has a ChIP-seq primary peak there. The last one to consider is peak to all, where you just consider all interactions, right? And so there are some pluses and minuses to these approaches. So when you do peak to peak, the number of interacting bends is quite small. right? So any signal that you have has a screaming high p-value because the number of interactions that you're considering is quite low. When you do peak to non-peak, the number of possible interacting bends goes up. Therefore, you need more read support to be able to say whether or not a interaction is significant from that modeled curve. If you go peak to all, which is something that you would do for something like HDK4 trimethyl, you know, it's the most possible bends that you could consider when doing HiChIP data. So you're really going to need a lot more data and a lot more read support per bend to really determine if those interactions are significant, because you have just a lot more interactions to consider when you're building those models. So now that we know what contact arcs are, and we know what primary and secondary peaks are, and we know how we call a significant interaction, let's go about doing that. There are a number of ways that you could go about this. You could do bespoke analyses if you want to. There are other prepackaged tools that will do this for us. I have this broken down by the tool where you could go and download that tool, the required input format for the HiChIP data, and whether or not there's an option for resolution, and whether or not there's an option to select your interaction type. So fit HiChIP is my personal favorite and what we'll be looking at today. It's my favorite because it is so flexible, and the math behind it is very, very, very simple to understand, and the outputs are really flexible. And not only that, it allows us to flag what resolution we want to look at and ask the question about what type of interactions we want to build our model around. There is C-loops, which works quite well. It takes an alignment and bed PE format, but it's very canned in that it doesn't let you select options for resolution or consider different interaction types. There's HiChIPper, requires HiC Pro valid pairs format, has a resolution option, but doesn't really allow us to select what interactions we want to consider. And then there's HiCups. This is really developed for HiC contact matrices, but it can detect things like CTCF HiChIP loops. And you can go that approach, but it was really developed on genome-wide contact matrices and HiC data. So the data we're going to use today in the fit HiChIP example is a Dovetail HiChIP MNase library on GM12878 that's been aligned to the reference genome HD38. So this is two pooled libraries. The total number of read pairs here was 478 million. Of that, 300 million about were no dupe read pairs. Of those no dupe read pairs, 241 million of those were occurring in cis-base, so that's about 80% of the good data of the no dupe read pairs. We have about 19% in trans-base, and then we have about 100 million valid read pairs that are greater than 1 KB to consider here. And you can go in and download these FASTQs off our Read the Docs page, and everything that I'm going to show you today here in the tutorial is also hosted on the Read the Docs page if you want to walk through it yourself there. So getting to identifying those significant interactions that I just talked about. So the questions that I'm really going to be asking here as I do this, this analysis is, how many CTCF interactions can be detected? Meaning, how many CTCF chromatin loops can I see with this approach? And then I'm going to ask about a region of interest. So what are the CTCF-directed interactions at the housekeeping gene RAB7A, right? Just a standard housekeeping gene should be on. So let's look at what the topology is, specifically focused on CTCF-mediated events. So for this analysis, we're going to use fit HiChIP. We're going to need our HiChIP data in valid pairs format, and then we'll need our ChIP-seq peaks in either a bed or .narrow peak format. So the steps here is we're going to prepare the data. So we're going to take the pairs that come out of pair tools. We'll filter for good pairs, then we'll convert that into HiChIP Pro format. Then we'll compare the config file, then we'll run fit HiChIP using Docker, and then we'll plot. And then I'll walk you through the 2 ways to plot. So first things first, how do we select good pairs? Well, pair tools is really nice for us, and it flags each pair with a specific read type. And so all we need to do is use pair tools select, and then list out the type of good pairs that are in that data. So once we do that, we feed in, we want to select all of these pairs out of our gzip pairs file. And then we will spit an output out of those filtered pairs again in gzip format. Then what we're going to do is simply using a combination of, that's awk, not os, of grep and awk, we will unzip the CTCF pairs file. We're going to remove anything that's been commented out. That's the £. I just aged myself, or hashtag, whatever you prefer. And then we're going to use awk, not AWS, AWK-F. And so we're going to print out a tab separated file where we're just going to select the certain columns of the pair tools format to then print out as a HiChIP Pro pairs format. And so we're just going to then gzip that back up, and then we're going to call this HiChIP Pro valid pairs.gz. All right. So now we have our good pairs in HiChIP Pro format. Let's move on to the configuration file. I know this is really small. On our read the docs page, it's much bigger. So I highly recommend you go check that out there. So you can open this in something like BBEdit, or if you're on the command line, you can use Touch, Nano, VI, whatever your favorite text editor is. And the key things here that we want to adjust are our path to our valid pairs file, which is right here. So you just feed in that path. Then what we want to do is feed in our ChIP-seq peaks. So here, peak file, that's the narrow peak format, or just a 3-column bed if you have a consensus file of peaks that you want to ask that question about. Then we want to point to where the output directory goes. Then we want to consider what type of interaction we're looking at. So here I have it listed out. So if you select 1, it's peak to peak, right, for CTCF, which is what we're going to use. There's peak to non-peak, something like POL2, something like peak to all, like H3K4 trimethyl, H3K27 acetylation, or all to all. It has an option to do this with HiC data, or you can select 5 and it just runs everything. It takes longer, but you get all the results that you want in one go. And then here we want to flag our bin size. And so this is a really important point here. Bin size is dependent on 2 things in this scenario. One is how much data do you have to build small enough bins to ask that question. The second is what is the size of your primary peak? If you have a very wide primary peak like POL2, you can get away with having a larger resolution because you're not going to span multiple peaks in one bin. If it's something nice and tight like CTCF, you want to be somewhere in between the 2.5 to 5 KB bin size. That's a good start for doing those analyses. So there's the one thing that you really want to consider here is that you don't want to have your bin size so big that you're capturing multiple primary peaks in a single bin. It can happen, but you want to avoid that as much as possible. So the bigger your bin size, the more primary peaks you're going to be capturing. It's not the end of the world, but if you can avoid it, you should. Then the next thing you want to consider is what is the distance that you expect things to interact with, which is the next flag here that we need to adjust. So the lower distance threshold, meaning I want to look at all interactions that are greater than X distance, is going to depend on the protein that you've pulled down on. Here, their default is 20 KB, which is quite long. And if you're thinking about something like CTCF, that makes a lot of sense actually to put a cutoff at that distance because you expect longer interactions. But if you're doing something like HDK27 acetylation, POL2, or transcription factors, these things often work in a little bit smaller of a distance. So you maybe want to shrink that depending on your question or your protein that you're interrogating here. One thing that you should keep in mind here is that the distance should be at least bin plus 1 bin, right? So you don't necessarily want to go to the nearest neighbor bin. You're going to get a lot of data that's just short inserts that aren't going to really be informative there. So at least have it 1 bin size away. So you're capturing those interactions there. So next with HiChIP, there's a few model parameters that you could build in. There's S, which is more stringent and really useful for peak-to-peak calling. It really gives you a nice tight model there. And so anything that's greater than that, the p-value there is quite strong. Things that are more noisy, like enhancers, promoters, transcription factors, you want to use the looser, less stringent thresholds. This is kind of because there are so much more data to consider that you want to really lax some of the statistical thresholds when you're assigning that p-value. It doesn't devalue that p-value, it just is more in line with the biological question that you're asking there. Moving to the end of the config file, the next thing that we want to do is flag what's our bias type. So there's a number of different biases out there. One of the best ones to use, in my opinion, is coverage, where you basically you normalize the contact counts per the coverage of your 2 bins, right? It's just saying, bin A has this amount of coverage, bin B has this amount of coverage. Let's normalize the contacts that are linking these two by that combined coverage signal. Or you can use an iterative approach that's known here as ICE. I typically don't see a big difference when using these 2 approaches. So I typically default to coverage. Again, it's a much simpler statistic to understand, and it really, we haven't seen it change the results in a fashion that really matters with the outcome. And then, so finally, the last things that we want to do here is flag our .genome file. So this is just a file, a text file of chromosome lengths. So it can build the bins, and then a prefix that you want to call everything. So now that we've gone through and edited the config file, now we just run. One of the reasons that I also really like HiChIP is that it's really flexible in how you run it. You can run it on the command line. You can run it via Docker. If you have sudoers privileges, if you don't have sudoers privileges and you're working on a larger server, it can also run on Singularity. And Singularity is essentially Docker for people who don't have sudoers privilege. So it kind of runs the same way. It's just a slightly different flavor. But the actual algorithm and the workflow is the exact same. It's just how do you manage the dependencies? And I find Docker to be one of the best ways to do that. So here, all we just really need is the config file, because we pointed that config file to all the other files that we need. And it's just simply run the fit HiChIP Docker shell script here, where we're going to flag the config file as what we've saved our configuration file as. And then you hit run, and it does its thing. It does its thing. It takes about, I don't know, 15 to 20 minutes, depending on how much data you have and what parameters that you've set it. So it actually goes pretty quick, all things considered. And then what you get at the end of the day there's a lot that's in there in the output, but one of the key things that you want to take a look at is the output. So what this does is it's a HTML file that will just load on your browser. And then you can look at, what your file was called. And then, basically what it's going to report here is the total number of cis interactions that match the parameters that we fed in. So this is what, 1, 2, 3, about 200 million, if I'm reading that right. Yeah, about 200 million interactions that it's trying to build a model out, or that you could possibly build a model out of. Now, for the peak-to-peak loops, it selects a subset of these possible interactions that fall within the condition that both bins fall over a primary peak. And then it asks, what is the number of interactions that pass a p-value threshold based on a false discovery rate, which is the FDR. So we're getting about 21,000 loops or interactions here that are passing that FDR threshold. Now, one thing to consider here is that oftentimes adjacent bins have the exact same interactome profile. And so what this does here is it actually merges bins that have the same looping structure that are next door to each other. And so after merging nearby loops, what you end up getting is about 17,000, 17,500, 600 interactions that all pass the false discovery rate. And so the plots that they're showing here are just what that model that they were trying to build that I showed you earlier. So here's that model. And then it's just giving you some information about those significant interactions. So how many interactions fall within a certain distance and then what the median is. And then what it reports here, it doesn't have a title, which is unfortunate. This is a number of interactions as a function of your p-value threshold here. And so what you can, it's kind of hard to see, but right here is our cutoff. And so it's telling us the number of loops. Oh, no, sorry, it's back here. This is our cutoff. So it's telling us the number of loops that happen at our predefined cutoff. Now, if we wanted to relax what that threshold is, all the way up to maybe say, so it started at 0.01, we could take this up to all the way up to 0.1. And what you can see here is that you would, yes, start getting more significant interactions, but they're dubious in their p-value. And so one way that I like to use this particular curve is if you, as you go up in your false discovery threshold, if this is plateaued or plateauing, you've pretty much saturated the number of interactions that you can find. If it's a very steep, if it's still increasing sharply, what that means is you really haven't saturated the possible interactions there to look at. And so there's two ways that you could go here. You either need to add more data, right, to be able to saturate those interactions, to be able to put a p-value on them, or you need to loosen your p-value threshold, understanding that you don't have enough data to be as stringent as you would like to be. So that's the two ways that you could play that. And either way is a good option. You just need to state, right, why you picked that route. Okay, so now that we have these significant interactions, how do we look at them? So let's plot them. So the first route that we're going to do is with R. And simply, we're going to ask the question here, what are the CTCF-directed interactions around this housekeeping gene? And so we're going to use R with the bioconductor package Sushi. It's super flexible, really easy to use. It's been around for a little while. It's a really intuitive tool. I recommend folks check it out if you haven't already. And then what we need to plot is those merged significant interactions as a bed format. So this number here that I've starred, it is in this file here. It's kind of buried in some subdirectories, but this is the file that you want to get, the merge near contacts.bed. And so the steps here is we're going to, first we're going to take our alignment file and then we'll convert that into a bed graph and a bigwig. And then we're going to load all of our data in R. Then we're going to do some very simple manipulations. So we have a column for distance, and then we're just going to plot those arcs in coverage. So first step, we're just going to use deep tools to convert our alignment to bed graph format. So this is simply BAM coverage of the deep tools. You feed in our BAM, our output format is bed graph. Our output file is the HiChIP coverage in bed graph format. We do the exact same, except our output format now is bigwig, and then we print out a bigwig file. And then we move to R for the fun stuff. So first we'll load the library. So you'll load this bioconductor package Sushi. And then you're going to load the data. So the coverage, we're just going to load the bed graph file. So we'll read.table, we'll feed in our bed graph. And then for the the arcs that we're going to draw, we're going to call it arc, and it's simply going to be a read.table. We'll feed in the fit HiChIP merge interaction contacts. And then we're just going to say that the header is true. The reason we do that is because there is a header in this file that is informative. There is not a header in this particular file that's informative, so we don't need to flag that. All right, and so what we want to do now is add a column for distance in our arc file, right? So really the structure here is just bin1, bin2, interaction count. There's some other information in there, but that's really what we need here. And so what we need to do is just add a column for distance. So we'll do this by taking the in position of bin2 and subtracting it from the start position of end1 or bin1. And so we'll just do arc. We'll add a column called dist for distance. And we're going to take the absolute value of arc anchor n2 subtracted by arc anchor column s1. So now we have a new column where each row has the distance of the bins that have that p-value that we want. Now all we want to do is set the region, the genomic region of where we want to print this plot out. So the RAB gene that we're going to look at occurs on chromosome 3, and it occurs within this window. So we should say chromosome 3 equals quotes. We have to use quotes here, CHR3, because it's a string, not a number. And then here for chromosome start, we'll list out in the position in bases, and then we'll list the end position out again in bases. And then to plot, it's actually really straightforward. So in Sushi, all it is plot bed graph to plot the coverage. We'll feed in the coverage file, and then we're going to tell it to plot on the chromosome that we flagged over the chromosome start to chromosome end region. And then here we're just adding in some, label the chromosome. Here's the text size. Here's what I want the axes to read and things like that. So, but the real crux here is this top line, which prints out your coverage. And there you go. Boom, you have your ChIP peak. Your coverage. So here are all of the anchors in the data. Then we want to plot the arcs. And so this is actually really simple in Sushi. So it's just simply plot bed PE. We're going to feed in our arc file. We want it to plot again over that same chromosome region. And so now what we want to do is flag out how we want to scale these arcs. So the arc height. And so I want to say the height is in the arc data frame, the column distance that we added. And then we want to make the plot type be a loop. And then we're going to flip it so it goes underneath the coverage. And this is what that resulting plot looks like. Now, this is really nice. If we already know that all of these pass the p-value, these loops look really nice and are easy to read. One of the things, though, that it doesn't really tell you is how many interactions are being captured by those two arcs. So like read support is not being captured here. So what you could do instead is, instead of scaling the height by distance, you could simply scale it by the sum of the contacts. So there's a column in the merge bed file that has the number of contacts that are linking the two bins. And so we can just say scale by the number of interactions. And so what this does is it doesn't change the anchor point, but it changes the height of the arc. And so what we can see here is this particular interaction between these two peaks here is really strong. And same here with this peak and this peak out here. Those are really strong interactions. And these are kind of weaker interactions because they don't have much read support. They are significant because they pass the p-value threshold, but they're still not as strong of an interaction. So that's one way in which you could look at these arcs. The next way is through the WashU Epigenome browser. This is a really great tool. If you're not familiar with it, I highly recommend it. So what you need here to get started is the merge significant interactions as a bed GZ and a significant interaction index that ends in .tbi. What's really great about fit HiChIP is it actually spits both of these files out, called they're flagged specifically as WashU compatible. So you don't have to do any formatting. You can just take these files directly into the WashU Genome browser. And then finally, we just want our coverage in bigwig format so we can plot the coverage and see how the peaks interact. So we'll go to the WashU Epigenome browser. We'll click Explore the new browser. We'll select Human because that's what we're working on. HD38 because that's where we aligned. Then you click go. And then what you'll do is you'll go to track list, and then I typically remove all the tracks that are there just so I have a blank canvas to start from. And then I go back to track, go local track. If you've downloaded them, you could go to server if you have them stored on a server. And then so first you'll load the bigwig. So you'll select bigwig, you'll feed it your bigwig file. And then to plot these arcs, you'll need to go and select the long range data type. And then you need to feed in at the same time, just select both the bed file and the index of that bed file in GZIP format, and then hit OK, and you're off and running. But what you can see here is you can see the coverage, but you can't see the arcs underneath. This is because it's default for long range plots as a heat map, and these are pretty sparse heat maps. So what you need to do is right click on that track, and then for display mode, just select arc, and then hit OK, and then go to your region of interest, type in the gene that we're interested in, and then hit go. And then it will report this plot out where now we can see the coverage and we can see the arcs, and it highlights the gene that we've just typed in. So a lot easier to manage than R, but I think R produces kind of more publishable ready figures. So back to the question at hand, why I made all of these plots in the first place. So what's going on with CTCF directed topology at RAV7A? And what we can see here, here's the RAV gene. What we can see here is there's an internal arc and an external arc that flanks this gene position. And so what we can say here is that there are two hierarchical interactions that are occurring with regards to this housekeeping gene with very strong boundaries. So one of the nicer things that this does here is the opacity of the arc is reflective of the p-value. And so these are really strong arcs here as compared to these, which are kind of weaker interactions. So these are really strong CTCF boundaries here. This is likely reflective of a sub-TAD-TAD format or a TAD loop of type structure. The other thing we can see here is that there is a weak interaction with this internal boundary to this external one and vice versa, which makes me think it's more of an internal TAD structure than anything else. But anyway, that's how you go about interrogating regions of interest once you've generated your significant interactions. So I do want to advise here that boiling down those contact matrix approaches to HiChIP and these contact arcs is really nice. And it's really nice because it gives us a reason to go look at a specific site, because we have a p-value that we can say, yep, this is important. But what's missing there is some of the subtleties that are being captured in the HiChIP data. So here what I've plotted is the exact same coverage plot that you see here. And this contact matrix has actually been distilled down into significant interactions. This is for H3K 4 trimethyl, right? So for active promoters. And if you just look at the arcs alone here, you can see that there's a lot of interactions going on here. There's a very strong interaction that's occurring here, but it doesn't really tell us a whole lot. Now, if we go back to these regions where we've assigned p-values, which is nice. So now you can say we have a p-value, let's go look at them. We can see a lot more complex structure here, right? So we can see some of these long tails that are occurring. And one of the things that is really interesting here is that there is a hypothesis out there that H3K4 trimethyl actually kind of directs pole 2 to read in certain directions. So the directionality of how these contacts go in the matrix are informative of the biology. So it's nice to have a p-value on them, but if you're just plotting the arc, you're going to miss some of that subtlety. And it is almost a little bit of an art form, and you need a lot of supporting data to make those claims. But it's, you know, you boil it down to a p-value, now bring that p-value back to the larger picture of what this protein-directed topology can be doing, which I think is the more fun question to ask after you get the p-value. All right. So just to end, we do have quite a bit of questions about what happens if I don't have ChIP-seq data for my experiment. Can I use HiChIP data to call peaks? The answer is yes, you can. It might not be the most advisable thing, and I'll show you why, but it's still doable. It shouldn't stop you. It just is a little bit more challenging. So the first thing that we need to do in order to do this is to call peaks. And so what we'll need is our alignment, and then we'll need SAM tools and max. So one of the things to keep in mind here is that max has a built-in assumption that your data are ChIP-seq data, and that the insert size of your read pair is less than 1 KB, right? Because that's how you make ChIP-seq libraries. With HiChIP libraries, read 1 falls within that anchor, but read 2, like I mentioned, falls someplace out in topology space. And so it doesn't like that really long distance between your 2 read pairs, so it ends up throwing out a lot of data, or it won't even run just because it says the insert size is too big. So the way to get around this is we're going to prepare the alignment by selecting just the primary alignment. So this means that when you align, you have a primary, secondary, supplemental alignments and all that. Here, you're just going to take the primary alignment, the best alignment that you can get out of those read pairs, and only take that one region and kind of feed it in as a bed file. So let's do that. So let's extract the primary alignment and convert to a bed file. And we'll do this with SAM tools and bed tools. So with SAM tools, it's view. We're going to filter for reads that have the flag 0x900. This is the flag for the primary alignment in the BAM file. So any primary alignment has this nomenclature associated with it. And then we're going to take that directly from our alignment. And then we're going to pipe this directly into bed tools, BAM to bed. Here we flag in the standard in, which basically is saying from a piped command, it's taking what the standard input is from that BAM file. You don't need to think about it too much. All it's doing is saying, take the input that's already been input. So that's the BAM file. And then we're going to convert that to a bed, and then we'll just have it report the prefix primary alignment dot bed. And then we'll simply use max2, call peak. We'll feed in our treatment as the bed file we just generated with the prefix with the end of mac2. And that will return, that will allow us to call peaks. But there are a number of things that you want to consider when you do 1D peak calling with HiChIP libraries. Data in blacklisted regions of the genome will be called, and they will normally show up as outliners, like they'll be screaming high. Blacklisted regions are typically regions of high repeats, telomeres, centromeres, things like that. You typically want to remove those from your alignment before calling, simply because you're going to have screaming high signal there where there shouldn't be. The other thing to consider is that only a fraction of your reads will fall within the DNA protein binding site. Remember that only one part of the read pair maps to the anchor. The second part is someplace off in topology space. So you actually need quite a bit more data than you would with ChIP-seq data to call peaks. And so this has 2 ramifications. The first one being that you'll be calling both primary and secondary peaks if you only feed in HiChIP data. So with your secondary peak, they're typically lower in size, and so that kind of contributes to your background noise. And because your secondary peaks aren't nearly as concentrated as your primary peaks, that background signal is high and it's pretty evenly distributed across the genome. So your signal to noise ratio, which is what max2 uses to call those peaks, can choke on that. So if you're trying to peak call with like 10 million read pairs like you normally would with ChIP-seq data, you're not going to get very good peaks, only because you maybe only have 4 or 5 million read pairs that are going into that. And of that, it only takes the primary alignment. And if your primary alignment occurs in the secondary peak, it kind of muddies that signal up. So if you're sequencing these data to do HiChIP analysis, There's plenty of data to peak call, so don't worry about that. But it's just more than you would be sequencing for a ChIP-seq experiment. Another thing that our approach here at Dovetail is something to consider is that we use MNase to fragment the genome. And so what that means is not only do we grab the DNA protein binding site, we grab the nearby nucleosomes. And so this will reflect in broader peaks if you just use that data. So here is sonnecated-based ChIP-seq data. And then here's MNase-based HiChIP data. And what you can see here is the peak for the ChIP-seq is quite high from the noise. And there is noise, but it dissipates rather quickly as you move away from the peak center. With MNase, here's the peak center. That's what a zinc finger protein binds. And then here are the nearby nucleosomes that are interacting with that zinc finger protein. And then we have more interactions that go further out. And so that background noise, it's not noise to HiChIP data because we want to know what nucleosomes are interacting with each other, but to max2, to peak call, they consider that noise. So you're going to get broader peaks there as well. So that's something to consider. Finally, something that's very typical to do is to aggregate your coverage signal around your peaks to know what the shape of your peak looks like on average throughout the genome. If you use ChIP-seq data and ask the question, what does the HiChIP coverage look like around ChIP-seq narrow peaks, you get a really nice ChIP signal, right? So you see here's the peak, here are the nucleosomes that are interacting on average with the average peak. Now, if we use the peaks that were derived from HiChIP coverage, the peak signal looks different. It's going to look wonky because you're capturing all of this signal and you're capturing the secondary interaction. So it's always best practices to use ChIP-seq when asking questions about the primary peaks. But if you don't have it, you still get a peak signal. It's just not maybe what your expectation was. And this is because you're asking a very fundamentally different questions, between primary peaks versus aggregating over primary and secondary and nearby nucleosomes and all that fun stuff. So I will end here on a few quick comments and things to keep in mind here. You always want to keep in mind, what's your question? What are you really asking of the data? Sort of like how I've highlighted here. What are you really asking when you aggregate using two different approaches to peak calling? Right, that fundamentally changes what your expectation of the output should look like. And then, even probably more importantly here is what's the biology? Why did you do this experiment? Why did you sequence to the depth that you did? Why are you asking, what is the biological question that you're really trying to drive at? And to that end, feature calling, p-value calling might not, be the answer that you need. One of our collaborators, they have so much ChIP-seq data that all they really needed was the HiChIP contact matrix. Don't even need to assign p-values. They just need to know what the interactome landscape looked like. They had so much supporting data that it got them into Nature Communications. It was a great piece of work, but they didn't have to do any real statistics on it because they had so much supporting data. So think about what it is that you need to know and what other data you have to support that. I kind of briefly mentioned this publish-ready figures. So GUI and server-based approaches to data visualization are phenomenal. They're great. They're really good for exploring in real time, for taking snapshots of region of interest, but they have real limitation when it comes to aggregating things, building averages, or really even building just publishable figures, right? These are often figures that I think you make and then you take to your PI, and then you and your PI sit down and look at them, and then you come up with what's the best approach to actually plot that. Most of what I do typically is generated in R. You can do it in MATLAB or Python if you like that route, but I just prefer R. Resolution, I like to think of it as like you're flying in a plane, right? When you're at 30,000 feet and you're maybe looking at things at 1 megabase resolution, you can see all of the mountainscape, but you can't see any of the houses and the cars that are on those mountains. If you fly really low down to like 1 KB resolution, you can see all the houses and cars, but you might only be able to see 1 or 2 mountains that are directly in front of you and to the side of you. right? You won't be able to see the whole picture. So call it multiple resolutions and see what you get. Here are just some file definitions that you can look at the end when on your own time, just what's common, what's proximity ligation unique. And here are all the tools that I've used in this workshop and the links to go get them. And I will end by saying thank you very much, and I will take any questions that I can in the remaining time.